Okay, now the today class for the people who are online. I want to apply for a microarray analysis, and I will try to explain the best I can, given that the author of the experiment is not here. But basically, this is an experiment done by Atakan on the expression of microRNA. So this is a microarray, an asymmetric thing, with oligos designed for some microRNA that's been identified in literature. It's something. It's a commercial platform, so it's well defined. You find it online. Is well established. So at the end of the day, what we get from the process, you get a number for each microRNA. If each mRNA, you get a number which corresponds to the expression. If that, in theory, corresponds to the concentration of the things. Okay, in theory, when we talk about normalization, you will see that that is not necessarily the case. But in principle, bigger number mean higher concentration, lower number mean lower concentration. Okay. Now, since I learned microarrays for gene expression, you will find me saying gene expression when I should say microRNA expression. And in fact, I had an interesting conversation the other day with the biologists that, in my opinion, everything that is expressed is a gene, but apparently not everybody thinks the same. Because not all genes are encoding, but they are still expressed. But anyway, whatever. So expression is concentration of something. I am not a biologist, so I may be wrong, probably wrong. So the data will look like this. In the rows, this time we have genes, and in the columns, we have the different arrays. The technical detail, the output of the scanner are a format called cell, that is typical for asymmetrics. So here, every array is a column. There is some, during the normalization process, we make this matrix. And the labels here are the following. The first word represents the diet. Okay, these are a mouse under different diet. L means ad libitum, meaning they eat freely. They are in I don't know, buffet, free buffet. They eat until they cannot eat anymore. They're happy life, happy. They are the happy mouse. These are the suffering mouse. The CCR, the calorie restriction, constant calorie restriction. These are the they are starving, and they look at the other mouse eating, and they are suffering, and they want to die. Fortunately, they die. In fact, after 50 weeks, this W means week 50. There are different times. In this case, they took after 10 weeks, after 15 weeks, and after 80 weeks. Okay, they kill the animal because the poor animal was suffering, starving so much. Then this T is the particular animal. It's the replica name. Okay, it's the name of the replica. So we have, and as you see, we have tissue. Tissue. In this case, there are 3 tissues: blood, brain, and breast, mammary tissue. Well, I didn't print all of them, but these are the conditions: tissue, time, and diet. Okay. And in fact, we have replica. You see here 95, 96, 97 are 3 different mouse in the same condition. Okay. 3 biological replicas for every condition. So, for this example, I'm taking the tissue and time are fixed. Okay, I will show you the simple case. So the only factor here is diet for this first exercise. Okay. Later, I'm going to make it harder. The condition is a factor. Sorry, let's say, so these are the column names that I show you. So you see that we have 3 ad libitum, 3 calorie restriction, and they have intermittent calorie restriction. Essentially, the difference is like these are like 3 weeks of full food and 1 week of not eating, and the other is 1 week of not eating and 3 weeks of full. So essentially the same, but essentially the animal is sacrificed at the beginning of the cycle or at the end of the cycle. Okay, it's like I don't know, if your last week of life was a good week that you eat everything, or it was a bad week that you were starving. So to make the analysis, it makes sense that we will have a factor describing this column, which in this case I made manually. Later I realized that I can do essentially process this data and get it easy. But here factor with well, you see what happened. We have 3 times ad libitum, 3 times CCR. Okay, everybody follow what I'm doing here. Now I have 12 values, and they correspond to the conditions, 1 for every column. Okay, so I'm forgetting all the detail about the replica, about the tissue, about the time or age, about the data format, all the things. The only thing that we care at this level is the diet. Okay. If I take the first gene, I can do the same thing that we did previously. I can make a linear model of the first gene, and we will have, by default, if you write this formula in R, it will assume that there is an intercept, and then the conditions are a comparison with the intercept, you know. And here it is important to know that the levels, unless I say the contrary, the levels are sorted alphabetically, and the first level is the reference. Okay, which in this case is what I want. In this case, for biology, I mean, given the way that we decided to model, in this case, ad libitum is the reference, and then every diet, like calorie restriction, are comparing with ad libitum. Okay, so if I do this model, I have essentially 2 kinds of value. First is the intercept, which is the average for ad libitum, which in fact doesn't tell me anything, because we know that the gene is expressed, but we don't know if that expression is a lot or a little. We don't have any reference. Okay, but what we do have in the other factors, this number is how much the gene expression, the Myrna expression, changed with respect to ad libitum. So when the condition is calorie restriction, you see in all conditions this gene is going down. Okay, the expression of this Myrna is lower in every diet, is a lot lower in constant calorie restriction, not so much in this intermittent, and then again a lot in CCR. Okay, of course, to evaluate if these numbers are statistically significant, we need to see the statistic, and with the linear model we can do that. We discussed this thing. Okay, just wanted to remind you how the things are done. Okay, fine. Okay, then we discussed last time that this is really done with a matrix with 1s and 0, when the 1s encode for every, they are 1 when the condition is true and 0 when they are false. And we can do it manually, and sometimes we do it manually. Sometimes, I mean, if you have a very specific condition, it is fine if you make your own matrices, but R can help you. In fact, what R is doing when you write the formula, internally use this command called model matrix that give you the matrix. There are other details here that are not important, but you see, we have a constant for the intercept, because essentially the intercept means that it's always on. And then a flag that is 1 when the condition is calorie restriction, and 0 in the others. Then another flag for intermittent calorie restriction, and so on. Okay? This is the matrix that we discussed last time that we can make. And here, essentially, you see that in the first three cases, when you have ad libitum, the only value that you have on one is the intercept. That essentially means that the intercept is going to be the average of the ad libitum condition. And here you see that the value that you are going to get for condition CCR is the sum of ad libitum plus CCR. Okay, no, I'm not saying, no, I'm saying wrong. The value that we predict for CCR is the sum of the ad libitum plus this delta. We do the system of equation that I did last time, and you essentially will find that this condition will be ad libitum minus CCR, and you will see if that is going up or down. Okay? This is just a reminder, just to make sure that everybody understands the same. If for some reason we don't want to have an intercept, for example, if instead of the differences, I want to see the absolute values of each case, I just need to add a 0 here. The 0 in this format, in this syntax of R, means don't use intercept. And in this case, you see the values are all averages, and you have to calculate the difference on your own. You have to do the difference manually, which is possible, but not necessarily a good idea. And then there is also the corresponding matrix, and you see the corresponding matrix is kind of very easy, is 1 when the condition is true and 0 when the condition is false. We're going to use this later, okay? I am just reminding you that every formula that we write here, notice that the important is the second part. The first part is not important here. The second part of the formula is represented by a matrix. You can make that matrix manually, or you can ask R to do it for you, and we will. We can save this in a variable that we call design, and after we, let me see what we have here. Okay, after we design it, it is wise to change the names of this. So here I put the columns to the column name, the column of the condition, the name of the condition. Ad libitum, CCR, ICCR, because I like it to have the short colors. Why this is useful? Because now I'm going to go to the microarray section. So, as you probably know, we analyze microarrays. Well, one of the most common tools, in particular the one used by NCBI, is called LIMA. Okay, which means linear model for microarrays, or for gene expression at this time. Right now, they are also doing RNA-seq, and you can do, and we will do all RNA-seq sequencing. Okay, but the logic is the same. And if you don't want to use R and you have your own package, you probably have something similar. But you will see here what is the advantage. The basic advantage is that when you have the design matrix, you can apply not only to the first gene, but you can apply to all genes in the same step. Okay? You can efficiently do a linear model for each gene independently. Okay? But in one step. And the basic reason is because it's just some matrix multiplication, and if instead of having one row, you have 10,000 rows, it's just a bigger matrix, but the mathematics is the same, and the computer can do it very efficiently. They are super in parallel, so super good. So, library Lima has the following ritual that we're, oh no, okay, ritual is a bad name, but the following steps. The first step is, we have the, remember, this norm data was one row for every gene, one column for every condition, okay? Then the design, one row for every condition, one row for every array. Maybe I should say again. Let's go carefully to the beginning. The original data, one more. The original data is one row for every gene or Myrna, okay? In this case, there are 1,900. We will check it later. Let's say 2,000 Myrnas associated with mouse in this array. One column for every array, for every experiment, for every hybridization. Okay? Every sample, if you like. In this case, we have 12 that are identified here. So 2,000 rows, 12 columns, one column for every array. And since they are replicas, we use a factor to identify the replicas. And they essentially call it like this, or more precisely, something like this. Ad libitum, CCR, ad libitum, calorie restriction, intermittent calorie restriction. So we have 12 rows, one for every sample, one for every array, and 4 columns, one for every biological condition, or treatment, if you like. Okay? Or if you have a wild type versus mutant, or whatever is the biological condition that we are testing. This is the biology that we are extracting from the thing. Okay? Notice that here we are only talking about diet. Later we are making more complex with time and tissue. Okay? Okay, so the point of Lima is that they have a couple of clever ideas. The first idea is that if you have the design matrix and you have the expression data, so essentially the rows of one are the columns of the other, so there is some kind of matrix multiplication that we can discuss later that give you in one step all the things that you want. And it gets a complex object that I don't want to waste too much time, because to be honest, most of the time we never look what is inside. You can, if you are very curious to know what is the, I don't know, standard deviation and scale and the degrees of freedom. There are some technical details, but we don't take this directly because there is the second clever idea of Lima. They have a model to estimate the variance. Remember that all things that we care at the end is to estimate the variances. Because if you know the variances, you know the standard errors, and therefore you know the margin of error, you know the p-values. All these things depend on the variances. So I will say again, something I say every time. You are not looking for average, you are looking for variances. Okay? And you need the average to calculate the variances, but the thing that you want to measure is a variance, not an average. That is what you want to measure. So here they have an assumption. I really don't know the details, but according to the paper, they essentially say, look, we have all these probes in the same array, and they are made together. So they are making the assumption that they have not necessarily the same variance. The noise that they affect them is not necessarily identical, but it's similar. So they are using all the data in order to estimate the variance. Okay? Not only all the conditions for one gene, like we did in a linear model for one gene, but also all the genes. Okay? And that is a clever idea, in my opinion, because as we always say, we need a lot of replicas to evaluate these things. And what they are doing is based on some hypothesis. There are some parameters here, like, for example, they assume that most of the genes are not differentially expressed. Maybe that is the point. The big assumption here is that most of the genes or microRNA are not differentially expressed. So most of them are around zero, and whatever you see around zero is just noise. Okay? So you can use that assumption to say, okay, most of them are really zero, and the variability that we see in most of them is the noise of the system. Therefore, we can use all of them to evaluate the variance. Okay? Are you with me here? So you will see in most of the cases we do these two steps together. For now, There is a third step later, but for now we got. So the eBase essentially take this fit object and give it back with more things that we are not going to see immediately, but you see there is something called T, something called F, the F value, the T value, the P value, the FP value. It essentially added all the statistical tests, okay? The e-bias is the part that do the statistic. The same way that in linear models, when we calculate the linear model, we don't get the statistic. When we do summary of the linear model, we get the statistics, okay? Again, we don't look at them directly. What we do, we use a function called top table. Top table take the output of empirical bias and give you the top 10 genes, okay? And what we have here? Well, one row for every gene, one column for every condition. Remember the sign matrix. The sign matrix have ones in the first column. Therefore, what we get is the average expression for ad libitum. Like before, this is the average expression of ad libitum. And here we have a caloric restriction versus ad libitum. This is the differential expression. Differential expression of intermittent versus ad libitum. I should have made us, but imagine this is what we call a star design. We have ad libitum in the center, and then we have everything compared with ad libitum. Okay, maybe I should draw it. I will draw it later. Okay. If I find my okay, I have to find the markers. So this is essentially the same thing, and if we look at the correct gene, we will probably find the same values that we did with the linear model, but not only for one gene, but for all genes at the same time. Then we have the average expression under all conditions, okay, average in total, okay? The average through all the matrix. And we have an F value for the ANOVA. What is the ANOVA asking? The ANOVA is asking, are all these values different from 0? Sorry, no. Are all these values 0? I'm not sure if they take the first one, but essentially the question is, are all these values identical to 0? If the probability is small, you say, aha. It seems like one of these numbers is different from 0. Okay? Or more exactly, the probability that you get these numbers when the real values are 0 is super low. Okay? Which is the typical question that you do. Okay, a couple of things here. You can, of course, focus on a particular condition. So in this case, I can add a option indicating which coefficient I am looking for. In this case, the second coefficient, the calorie restriction. And we have two values for this one, the log fold change and the average expression. Okay? These are sort of different because the power values change. We have the t value. First thing, instead of ANOVA, we have a t value because it's one condition. Okay. For the t value, we have a p value, and for the p value, we also have an adjusted p value. Okay. I don't know how much I need to explain the adjusted p value, but one day I will show it. But essentially, the point is that every time you do a test more than one time, you have to pay a price for that. You know, no? I mean, since the p-values is essentially telling what is the chance that you get this number just by randomness. Well, if you do that again and again, just by randomness, you will get a false positive because you keep doing it. Each one can be small, but if you keep doing, Sooner or later, you will have one false positive. And so to avoid that thing, there are a couple of strategies to adjust your p-values, essentially not to fool ourselves. And there is a B column that I am, to be honest, not completely sure what it means. I mean, they have a definition, but if I try, it doesn't work. If something is not exactly, okay, but it's something like the log of the p-value, okay? Not exactly, but just to get an idea. The smaller the p-value, this more positive this number will be. And in particular, if they are more than 0, that is good. Okay? Any question? Okay, then you may realize that these are the top 10 genes and the white 10. White 10. Well, it's just the default to show something, but you can in fact say how many numbers you want to show. And what I normally do is say that the limit number is infinity. Show me everything. In fact, the point is later you have other condition. In fact, I forgot to put it here. I normally can say, give me everything, but code with a p-value of 5% or 1%, and you can put a threshold of the p-value, and then it will stop at the corresponding p-value. Okay, but here I wanted to show everything. This is, of course, a huge table. Oop. It keeps going forever. Okay. Tamam? Move away. What else? Plot. There are a couple of plots that, in fact, I wanted to show you because I saw them explain it. Let me. Okay, in a minute I will show you this in the NCBI webpage. They explain them. I am essentially showing how they do the plot that they show. Here, again, I am choosing one particular coefficient, and the input is the statistical test, the empirical bias. And for this coefficient, the calorie restriction, we have this analysis. First, let's say in this line 0 represent no false change. If the numbers are positive, it means that the relative expression increases. This is a gene. Each point is a gene, okay? And each gene has two values. One, if it was going up or down in this plot. And second, what was the average expression that they have? Like, was this gene super active or not active? Okay? Maybe, well, one of the points is that you realize that the genes that have low expression, they also have low average expression naturally. If they are 0.1 in every condition, then the average will be 0.1, and the average cannot be very low if one of them is very high. So the average expression is the horizontal, and you essentially see this thing. You will see later, this tend to be longer. Okay, here for this particular experiment, most of them are low, but some of them are very high. Point is, they are very high in all conditions. That doesn't change between the ad libitum and calorie restriction. Okay. Then it is interesting to show the p-value versus log-fold change, which I did manually here. Okay. You will see that's a common thing. Small p-values are good, high p-values are bad, log-fold. Well, so the point is that this we really don't care very much about this. So what people do, instead of plotting the p-value, they plot the log of the p-value. They plot log 10 because sometimes it's easy to understand. 0 here means that the p-value is 1, very bad. -1 means 10%, minus 2 means 1%, minus 3 means 0.1%. Log 10 essentially count the number of zeros. Okay? And this is almost good, except that it's kind of upside down because the good numbers are down, so, and you have a negative. So people say, okay, look, I know that these numbers are always going to be negative, and I really care about the most negative ones. So let's draw it upside down, minus log. Okay? Minus log 10. Minus log 10 p-value. And this is called a volcano plot. Okay, because it looks like a volcano. And it's so common that there is a function in Lima called volcano plot. You see, let me show you. Before, plot minus 10 log p value, blah, 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 blah. Forget about that. Volcano plot. Okay, do everything for you. Now here is the interesting thing. In fact, I should, well, I have one thing, but I think I should have done a couple of other things. One thing that we do here is we put a threshold for the p-value. Here I put a line in 5%. So essentially, everything that is below this horizontal line means that they don't pass the statistical significance test. Okay? So only points over this limit have, well, you have to define the statistical significance. Maybe 5% is too high. Maybe you want to be more strict and you want to have 1%, and you will put the threshold here. So, but the ones that are here are the most significant. And in fact, I forgot, but there is an option here that you can say, show me the best, the name of the best genes. And you put a number and you get the number here. Okay? Then we well, no, before going to the more conditions, the other thing is see, this number here, these genes here are statistically significant from 0. So with some confidence, we know that this gene is there not for noise, not for randomness, but for a real reason. Now, the change of expression between 0 and, I don't know, 0.1 is important or not important? From the statistical point of view, the only thing that I can say for sure is that this number is not 0. It's different from 0. But it may not have any effect. Let's say that I don't know, this microRNA is interact with something. How much should change the concentration of this miRNA to have an effect? How much will it change? I don't know. For example, I always have this in my mind. If the gene is an enzyme, increasing the enzyme concentration maybe will not have, or maybe worse, it's a structural thing like a membrane transport or something. Making more of that gene will not necessarily have an effect in the short term. I mean, maybe 1% extra. Okay, genes that are housekeeping can vary in the order of 1% without having any effect. On the other side, if it's a transcription factor, a little change will encode for the protein that will bind and will trigger everything. So to me, it's super confusing. And when I speak with the experts, in fact, they say, we have no idea. So there are different rules. The higher, of course, the higher log fold chain, the more sure you are that they are significant. I will say that these three here certainly have a good statistical and good biological reason to be interesting. But the one here are not clear. So some rule that I personally find that makes sense is to cut here with +1 - 1. Why +1 - 1? Because this fold chain is counting, how can I say, how many times you fold, you duplicate. So, okay, in other words, this essentially corresponds to the log in base 2. So the concentration, the log fold change of 1 essentially means that the concentration increases twice. A log fold change of -1 means that it reduces to half. And if something goes to twice the concentration, it essentially increases 100% or reduces to 50%, I think is clearly significant biologically. Okay. Some people may argue that the threshold can be lower, and yeah, maybe. But to me, 1 and -1 is something that kind of sounds correct. Of course, I am not a biologist, and the real biologists should be the people who can tell the correct answer here. Okay. Your job, in fact, will be to try to, before doing the experiment, try to find what is the change of concentration that you will consider to be biologically significant. Maybe for that we need to know the system, have kind of the pathways and something. Well, depending, of course, of the gene, but well, I will say that in the system biology can eventually help on that, the full system. Okay. Any questions so far?
I might have one.
Yeah.
Is this, so every point represents a gene here, but these
Yes.
are p-values. So as we know now, one or more of these genes are going to go below the threshold if we take the adjusted p-value then.
Yes.
So is this a bad idea or a good idea to show the adjusted p-value or the p-value plot? So which one should we interpret?
A very good question. To be honest, I always do this thing with adjusted p-value. I think the adjusted, well, there is a question how you adjust them. There is a couple of strategies, but in general, I go with the adjusted p-value. And to my surprise, this function is not using the adjusted, it's using the raw p-value. I show with the raw p-value just to show the idea of the Volcano plot, but maybe later we check the manual, maybe we have an option to use the adjusted p-value. And I think one of the reasons is because if you notice in the numbers that we got for this case were very bad. Let me show you. And that was the origin of our problem here. The adjusted p-values here are almost 1 in all cases, you see, 0.98. And the best one is 57%. So in this case, the adjusted p-value essentially say, look, you have nothing. Okay. So the p-values don't look so bad, but given that we are doing a lot of time and we have to adjust, this is nothing. And that is when, well, that essentially motivates the second part. Thank you. It will give me a good way to go to the next step. So yeah, when with Atacan we started analyzing this thing, we found that we had very few or maybe 0 genes that crossed the threshold, and we did a couple of things. One thing that we did, but it's already done here, is that instead of using all the microarray, we used only the oligos corresponding to mouse, because this is a big platform and include microRNA for human and for yeast, I don't know, many other organisms. And since we know that the experiment was done with mouse, we choose to forget the rest of the oligos, reduce the number of genes or probes, and that essentially reduce the number of multiple comparisons. Instead of comparing 10,000 things, I'm comparing 2,000 things. And that essentially make that the what is the punishment or the penalization by the multiple tests is less strict. Then the second thing was I say let's add more conditions. And let me see. Here I will show you this. I have not seen how it works. So now, with some effort, we got all the data, and you see here we have something similar, but here we see all the tissues and all the times. We have brain, week 80. We have well, calorie restriction, and after this you have ad libitum blood, week 50, and all the things. Okay. So in this case, 81, and there is something. Let me see if I find somewhere. There is something called, yeah, this one, baseline. So baseline is a sample taken at time 10 weeks for tissue, for every tissue. Okay, so the story is this: they grow the mouse from since they are born, and they are in the same condition until week number 10. At week number 10, some of them are sacrificed and they take the baseline, and the rest go to different diets. Okay, so baseline is the same for all of them, for all conditions, and this is a good reference. One of the problems with the previous analysis is that we arbitrarily choose ad libitum as a reference. This baseline makes more sense. You have at the beginning of time, before the diet intervention, you measure everything, and then you compare baseline versus ad libitum, baseline versus calorie restriction, and different times, and so on. Okay. Okay, how we did this thing? Just a summary. These are all the columns. Here are the baselines. There are 3 replicas of baseline in every tissue. Baseline depends on tissue. Okay. Then, with some magic that I wrote here, I essentially separate the names. Let me show you. So I noticed that first this .ga.cell or this .cell was just noise, so I drop it. Okay. Then I separated everything with this underscore. Okay. Well, something that I regret here is that the file name should have been more coherent. So one of the things that I always insist that be coherent in the way that you put name to your files. So the .cell was uppercase sometimes and was ga cell lowercase in other cases. Anyway, so these 2 commands here remove that extra, and here is split in 4 columns in a matrical feature that I later move as a data frame, and I put the column name so they make sense. So we have 4 columns: diet, replica, tissue, age, and for the baseline, there's no time. So maybe I should correct. I'm not sure if that is relevant or not. Okay, you follow me, no? Then I found another error. I see this biologist. Brain with big letters and brain with small letters is different, of course. So later I have to change the. So okay, I did this thing. I went to first I put everything in lowercase to solve that problem, and I make a design matrix with this formula: 0, because I don't want an intercept. There is no base condition that is common to all one. Okay, I say, okay, I am going to choose my own base condition, because as we say, the good base condition was the baseline, but it depends on the tissue. So for some tissues, I'm going to use baseline. I mean, for some sample, I'm going to use baseline blood. For other, I'm going to use baseline brain, and so on. So when I do this thing, I get this matrix here with names that are kind of long. Diet ad libitum age, a long name. So, and in fact, I keep going. And I got the ones in the zero. Well, so after some extra manipulation, I get the final design, which is this. I have here 81 rows, if I am not wrong. Yes. And I have ones only in the row that correspond to the sample, where I have baseline 10 weeks blood, okay? Or ad libitum 49 weeks blood, which happen to be 3 of each one. Remember, there are 3 replicas for every condition. So we have 2 tissues, 3 times, that give me 6 combinations, plus baseline 7 combinations. What I'm missing? 2 tissues 3 times, 4 diet. So yeah, 2 tissues 3 times 4 diet. The 2 times 3 is 6 times 4, 24, plus baseline 27 times 3 replicas, 81. That is essentially how the thing decomposes. You follow me? Again, 2 times 49 weeks and 81 weeks. Okay, those are the 2 times 49, 49 and 81. Besides baseline. Okay. So for each one of these, I have 3 tissues. So 2 times 3 tissues, they give me 6. Then 4 diets, that give me 24. Plus 3 baseline, that give me 27. And for each one of these, we have 3 replicas that give 81, which is the number here. Okay? Well, so eventually, we will apply the linear model over this design. And since there is no constant, what we will get is the average of every condition. So how we compare them? Okay, let me say it this way. The question that the linear model can answer you are always questions of the sense, is this number 0 or not? Okay, that is kind of the question that you want to ask. Is this number 0 or not? So you have to frame your question in that sense. And in this case, the question that I wanted to ask, at least at the beginning, is like, did the expression change from the baseline? So for this is the last thing that Lima provides us. We have a tool called Make Contrast. We take the design matrix and we describe what we want. So all this design matrix that I was kind of explaining with the system of equation, you can write it and Lima do it for you. So when I compare ad libitum at 49 weeks with the baseline, this is a minus here, so I will measure here the ad libitum minus the baseline. Okay? Caloric restriction minus baseline. And I create a new variable that I essentially put a D representing delta. So this is the chain of expression when you move from baseline to ad libitum 49 weeks in blood. Okay? Then similar for brain and similar for mammary tissue. Okay? And eventually, I think we have somewhere other option, like you could add other lines like comparing ad libitum with CCR. Let's say maybe I want to compare this with this. Directly, the baseline is the same, so they change. inside the same tissue, it makes sense to compare them directly, no? If you are comparing different tissues, you maybe need to add to also correct by the difference between the baselines. But the point is that we can do this description here, and I hope that you can find it easy to understand what we are saying here. I mean, like, I hope it makes sense to you by intuition. Okay? Please say no if you don't follow here. This is a critical. Here is when you explain what you want to measure. Okay? Here is when we move from what we measure in the experiment versus what we want to know. Okay? The biological question. I mean, the good experiments are, okay, let's say, a simple experiment is like I measure the temperature of a person. Okay, I put the thermometer here and I measure the temperature. And I get a number, 37. Is that good or bad? A number itself, the gene expression by itself, it will not tell you anything. It's a number. The interesting part is when you can have some value on that, you can assign some meaning. And here the meaning is like, what is the change of expression versus, I'm sorry, there is even another point that we have different source of noise, different source of error. And as I say, it is not obvious that the concentration, that the light intensity, the luminescence that you measure in the microarray, they are not obviously connected to gene expression. But the difference does. Because if there is anything affecting this part, will also affect this part. And when you compare them, all that noise cancel each other. Okay? So that is why when you see people studying differential gene expression, they always go with differential expression. Okay, because the comparing how things change, first it has biological meaning, and second, you correct for a lot of technical error. So this is a contrast that happened at the end, and you see the delta ad libitum 49 is 1 of ad libitum 49 and -1 of baseline blood. Okay, al 49 blood versus base 49. So eventually you realize that you can do it manually. These are all -1 in here. Well, I give you all the command. You can repeat this thing. I don't know if I can give you the data right now, but we can try. Okay, let me see if the owners of the data, yeah, they didn't show up today. Okay, now to be honest, there should be another slide that I didn't finish before the class, but I can finish now. So let's go directly to RStudio and we finish here. You see here I was doing the class. You see the class is here. You know that I do the slides in R. These are the slides. So I have the contrast and I have here my memory helper, how the thing was done. Here. So let me translate this to this. We are going to repeat the steps before. Okay, so we are the contrast. Let's do the fitting. So let me go back to what I say that they are the classical step. The first step was LM fit. This one, LM fit, normalize the data, the sign. This will be the first step. Okay, so the next slide is going to say fitting the model. I'm going to insert a chunk here and I'm going to say fit LM fit Norm data. Let me check. This is norm. No, this time I call it full data. The normalization data was the first example. This time we have full data. And I'm going to use matrix design. I am going to use the thing that we saw, okay? And then I'm going to do the second part, if you remember, was empirical bias. Well, no, wait. Should I do that? No, there is a step that I'm, okay, let me show you first how we did before, and then how it changed. Okay, let's do in 2 steps, okay? The second part was the empirical bias and the top table. Okay? Just follow the same protocol as before. And here I delete all this nonsense. So we do lmfit full data with the design. There's some warning here that I'm not going to. Okay, because there's something that I'm missing here. What is the sign here? No, 1 second. 1 second. I need to change the name of the contrast here. 1 second, okay? This is like in the TV shows about kitchen. I have the full pre-cooked. Okay, this is the same matrix. No. The point is that the column name had to correspond to the values in the data. No? Does it work now? Yes. So e-bias again and top table just to get an idea. Okay. So remember the design initially was only diet combined with age, combined with tissue, no intercept. So here we have baseline, the average of baseline, the average of radiatum. All the averages are around 11, you know, they're all averages. So these are all true things, but are not the question that we really want to ask. We are not really worried about what is the expression level in this condition. We are interested in the contrast between baseline and this condition. Okay, the change. And that is why we have the contrast. So there is a step that we do here that we call, if I am not wrong, let me see if I do it before or after. This contrast fit. Fit contrast, I say contrast fit, also part of Lima. And here I put the fit and the contrast. The contrast is the thing that is this one. Okay, this matrix that I show you. This one that I show you here. Okay, the matrix that essentially say how you have to compare things. No. This was the sign. Contrast. Let me contrast. Contrast matrix. Not this one. Oh my gosh, what is my contrast matrix? This contrast. Contrast. Contrasts. This one. Okay, every row is the thing that we calculated in the initial linear model, and every column are the different that we want to calculate. For example, here delta at Leviton 49 is at Leviton 49 minus baseline. Okay? And you can do more complicated. Here I went with something simple, but you can do more complicated. If you write the formula here, this matrix will change. And now the e-bias is over fit contrast. Let's do lmfit, fit, empirical bias, top table. Voila. You see now the top table gives you the deltas. This time are positive or negative. And at the end, you get the F value for the, well, the average, the F value for the, what is the name? ANOVA, p-value, and adjusted p-value. Okay? And we can ask for every particular coefficient. Well, let's say coefficient 2 like before. So, what was the coefficient 2? Well, we can do the Volcano plot. But the point is that now we have adjusted p-values that are better, no? How this happened? It happened because we used a longer model, and I'm essentially, instead of using the 12th column of the first part of the class, I'm using 81 columns to estimate the noise of every gene. Okay? And I can do that because I make a super big model together. So all the data is being used for the calculate the variance. And the initial linear model is super simple because the design was super simple. The design is like, well, let me just remind you what is the design. The design is nothing plus the diet plus a combination of diet, age, and tissue. Okay? So essentially, we measure the average of, we measure the average for every diet, every age, and every tissue. Every combination of diet, age, and tissue. That is the point. This formula essentially says take the average of every diet, of every combination of diet, age, and tissue. And that was the first model that we got here, no? Was before. This one. Every diet, age, and tissue, and this is the average. Okay, that was the initial model. And then the novelty here is that we did a trick with use contrast. And the contrast is this matrix that is multiplying the smart way. And when you include the contrast, the statistics, the numbers are different now, and the statistics are corrected according to that. And you get the thing. And then you can do the Volcano plot and all the things. You got me? Do you have any question? Yes, no, maybe? Panicked? No?
No, I don't.
Well, I have a question that I was, okay, tell me, Tunch.
I didn't quite catch how the change of the column numbers, or how many of the columns changed,
Okay.
which changed our p-value. How did that affect our p-value? So we had several columns. Now we have lesser columns. So that lessening the columns numbers affected our p-value, right? Did I understand?
Yeah, I mean, the point is that okay, let's go one by one, but essentially the p-values essentially answer the question, is this number different from 0 or not? In the first case, let me go in a couple of things. I am going to do a fake, so to have the comparison. I am going to do and statistic 0 here, you see, AB0 is going to be E-base, from the feed data before doing the contrast. And then I do the contrast. So I will compare before contrast and after contrast. Okay, is that your question?
Yeah.
Okay, so let's do. I can, and you see it's relatively fast. Well, just say that it got slow. Okay, good. So let's see one by one. So let's see the top table. Let me show the top table for the raw data. Okay, and maybe here, let me take a small number. I think I can take the first two, and will be the first two genes. Okay, doesn't need to be more to show what you are asking. So the column here are the names that we put in our design matrix. Here we only use the sign, and the design was essentially saying take the average for every combination of diet, age, and tissue. So you see all the numbers are a kind of big numbers, 11, 11.9, 12, 12.4. This is even a 13 here. All the numbers are big because the average of all these numbers, of all the gene expression are big. It's kind of the level that you measure, the measurement, okay? All are high and all are very similar. Then the question that the p-value asks here, this is a ANOVA. You get an F value, and this is ANOVA. The ANOVA is essentially asking if all the numbers are really zero in reality. If all the numbers were really zero, will we see what is the chance that we see these particular numbers if the real numbers are zero? And the probability is, how can I say, infinitesimal, no, minimal. But that is not a surprise because the numbers are big, because these numbers are not the difference, are just the absolute expression. So the p-values are beautiful, but are the answer to the wrong question. Now, instead of looking at the data from the original design, I'm going to look at the data from the contrast. Okay, this is the e-bias here, e-bias contrast, if you like. So I'm going to check the new thing. Now, the genes will be sorted different because, as you say, the p-value change. So since they were sorted by p-value, the genes that you see are completely different. If you force me, we can try to see the same genes, okay? But certainly the p-values are different because the best p-values before were 10 to minus 89, and now the best are 10 to minus 20. So everything should be different. And now you see that the numbers are not around 11 or 12. They are sometimes negative, sometimes positive, sometimes 1 point something. But if you or minus 1 point something, but not so big. And the F value is smaller and the P values are smaller. Okay. And these p-values are the p-values that kind of make sense. I mean, again, the p-value is answering the question if the real change of expression was zero. If the real values are zero, these are change of expression. So in other words, if this gene in reality never changes expression, in reality this gene never changes expression, what is the probability that we measure this number, assuming that, in fact, nothing is happening? If nothing is happening, what is the chance that we see this number? And the ANOVA tells you, look, the probabilities that you get this number when there is nothing happening is 10 to minus 20. In that condition, most people will say, yeah, I think the initial conditions are the real values are not 0. Something changed. Which one? Well, that is a different question, but a priori I will say this one. I think I can even choose the name here. I can even say top table, and here say coef. I can probably put the column name. And I get, well, I didn't get the same because it was not the best. But when you choose a particular column, a particular coefficient, you get the change of expression for that particular condition, and the t-test and a p-value for a different question, okay? So you have different values depending on the question that you are asking. If you don't specify coefficient, you are doing an ANOVA, which is testing all at the same time. If you do specify a coefficient, you're doing a t-test for that coefficient. Okay? So maybe instead of 2, I should have for the top 20 or something like that. Did I answer your question, or did I make more questions?
No, it actually answered my question, but born a new question. But I think I understand. What I understood was basically when we lowered the expression levels, numbers, to the 0, therefore the p-value goes up higher because the probability of the values of those are getting closer to 0.
I think you're saying it backwards. Okay, okay, maybe, I don't know, I'm not sure exactly what you're saying, but if the values are closer to 0, the p-values will be smaller, because remember, the p-value is essentially, okay, quotation mark, this is not strictly correct, but just to fix the idea, the p-value is like, what is the probability that this number is 0? If the number is high, the probability of being 0 is very bad. I say it wrong. What is the probability that this number is different from 0?
It went from -80 to -14, so.
From -80 to -20. So,
the p-value got higher, did not?
The p-value got higher. Okay, but I mean, but the point is that I don't get in, like, why are you comparing two things that are not comparable? Like, these two values, it's not that the p-value changed, it's that the question changed. Your question got a different p-value because it's a different question. It's like, what you are asking me first, what is the chance that it will rain tomorrow? And then you ask me, what is the chance that there will be an earthquake tomorrow? And my answer will give you a different number because I have different questions. Okay? In one question is like, what is the chance that the average expression is high? And the other is, what are the chances that the differential expression is high? Okay.
Thank you.
It's not that the p-value changed because we changed the number. We changed the question.
Okay, I was still thinking about the average. That's
No. That is essentially the point. Like in the first here, before I apply the contrast, I take the averages, and you see the names. They are all conditions. In the second, after I feed the contrast, I have the deltas.
Yeah. Okay. I missed the delta point.
I wanted to put Greek letters, but I didn't went to the level of putting. I mean, I don't know in window. Sometimes you can write Greek letter. I wanted just to show you something. I was looking here. I was choosing this gene, no, the 1521-9. And because in brain it was 1.6, and then I went to brain and I asked the top 20 of the top 20 of ad libitum brain, and I found the same gene. Yes, and the log4 change is 1.6 again, but the p-value is different because again it's a different question. In the big table, if the question is, are all differential expressions equal to 0? And you get a p-value. In the second, when I choose a coefficient, the question again changes, is this particular coefficient different from 0? Or is this particular condition? Did the expression change when you go from baseline to ad libitum? That is why you get different p-values. Okay, because, I mean, let me phrase it in a different way, because I remember having this discussion many times. When people present something, I did an experiment, I got a p-value of this. And I always say, well, what was the question that your p-value answered? And in too many cases, people don't know. People, they say, no, I use it SPSS. How you calculate the p-value? I use SPSS, or I use, I don't know, GraphPad. But that is not the answer. The answer is, what is the question that you are asking? So again, remember that the first class I say the first thing that you have to have clear is what is the question that you are asking. And eventually we will frame that question in the context of a question that a linear model can answer yes or no. And the yes or no will be a p-value or a confidence interval or something like that. Okay? Okay, the question that I didn't solve here, and because it's a little more kind of full of technical detail, is how to do contrast with the simple linear models. Because here I did everything in Lima for microarray. So if you are doing microarrays, let's say that the problem is solved. I mean, except the normalization. But essentially, if you have your data normalized, I think we discussed all the theory, and we just need to do some practice to do. Maybe we should do that. We should find some public data and ask people to do some experiment. But if your experiment is different, your experiment is like qPCR or something else that you are measuring, then the question that comes is how we do contrast with the basic linear model, this LM here. And I will ask you to let me answer that question next week, because I'm not completely sure yet. I tried something and it is a little tricky. I mean, I have a long answer, but I think next week I can have a shorter answer. Okay? Any question? Any other question? Yes, no, maybe. Sometimes. You know that I like questions, no? Okay, last point, just to show that I updated the slides, okay? This is the part that I wrote, and these are the results. Yeah, maybe I should write all this comment that we had in the meantime. Fine. Okay. See you next week then.